So today I'm going to be talking to you guys about hard clam restoration in Pine Island Sound, a pilot study. But first I want to just give you some background information and just kind of tell you what inspired me to submit a proposal for this project. So I started working for SCCF in January of 2019, and I think soon after that we did this scallop search in Pine Island Sound, and I was really excited about it because I could go snorkeling and look for scallops and seagrass, but we only found one scallop. That was very disappointing to me. So I went to my boss, Eric, and I said, Eric, why don't we do a scallop restoration? Scallops are so cool. They have all these eyes. They're so charismatic. I love them. And he said, it's just too hard to restore scallops with red's hide because scallops only live for 1 year. So then I thought back to the work that I had previously been doing. I was a research assistant at Stony Brook University, and we were doing hard clam restoration there. So back in the 1970s in Great South Bay, they had this beautiful, lush seagrass ecosystem, but then it got over-harvested. People came in and got all of the hard clams that were there, and it basically decimated the ecosystem. And they started getting really bad, harmful algal blooms because there was no clams there to filter the water, take up the nutrients, and all the seagrass died. So they did this huge hard clam restoration project, and it was actually fairly successful. So I just wanted to bring my ideas from that to here. Basically what they would do is they would take these big 18-wheeler trucks and fill them up with clams, and we would put them on boats and kind of just dump them out in the ocean. So that's kind of what our plan is for here. But first, we're doing a small pilot study to see if it works. The pictures that you see here are some pictures from snorkeling in Sanibel. This one is right by the causeway. So ever since I started this project, I've just been looking for hard clams because I like to snorkel when the water is clear. And there are actually some hard clams out there now. So it just goes to show that this project could potentially have some success. This one is by Bunch Beach in Fort Myers. And this one here is a picture from our collaborators in Sarasota Bay. So with this project, we actually started a new collaboration with other people who are doing hard clam restoration, which has been really wonderful. I've been learning a lot from them. For those of you who don't know what a hard clam is, I'm sure you guys have seen them when you're shelling, and you might even see them more now that you're going to learn so much about them, but they are basically the main ingredient in clam chowder, which is where most people might know them from. But while they're still alive, they can be found in seagrass ecosystems, either buried in the sand or on the surface, and they're just large, round, white shells with prominent ridges. They have a similar ecosystem role as oysters, so they're both filter feeders, and hard clams are really different because they can improve water flow by reducing seawater turbidity and filtering phytoplankton from the water. And they also increase the transportation of nutrients from the water to the benthos. And this is a huge benefit for seagrass in two ways. So first, they're taking out the phytoplankton and increasing the sunlight that reaches the bottom. And second, they're also reducing the competition for nutrients with phytoplankton when they remove the nutrients from the water column and put them down near the seagrass roots where they take up the nutrients. Another cool thing about hard clams is once you have like a really established population, the dead shells begin to accumulate on the bottom, and this creates structure, and that attracts marine life because the ocean is basically this big open water column, and once you put some structure in there, animals and plants, they start to get attracted to that. So this will increase biodiversity wherever it occurs, whenever there's an established population. They're also a prey item for whelks, crabs, skates, and rays. And so they transfer phytoplankton primary production to higher trophic levels. In the past, hard clams were abundant on the coast of Southwest Florida. We know this from evidence from the Calusa Indians in the form of shell mounds. So this is Mound Key in Estero Bay. This was actually an island that was formed by the Calusa Indians, and it was actually the capital of the Calusa Kingdom. Hard clams and other mollusks were consumed and then disposed of regularly in these large ceremonial events. And an archaeological dig in Mound Key actually found that the creation of the mound dated back to 300 to 500 AD. And they did this specifically using hard clam shells found at this site. So we know that hard clam populations have been established here for a very, very, very long time. In fact, the largest hard clam fisheries in the world in the early 20th century was in the 10,000 Islands, which is south of here. It was this 40-mile long, 5-mile wide swath of seagrass bed, beautiful seagrass bed, and it was thriving with hard clams and seagrass. It was very beautiful. But once people found that the hard clams were there, it got overfished, and the fishery collapsed in the mid 20th century due to overharvesting and also issues with water quality. All right, so time to get a little bit scientific here. There's two species of hard clams in Southwest Florida. There's the northern hard clam, Mercenaria mercenaria, and the southern hard clam, Mercenaria campechiensis. So the northern hard clam has a range from Nova Scotia and a little bit into the Gulf of Mexico. And the southern hard clam has a range from New Jersey and all throughout the Gulf of Mexico. The southern hard clam is the native species to this region, but the northern hard clams were introduced with aquaculture in 1998. Where aquaculture exists in the Gulf of Mexico, the northern and southern hard clams actually hybridize to produce fertile offspring. So there's this constant, they constantly are producing these northern hard clam genetics here, and it's kind of swamping out the southern hard clam genetics. And that's kind of one of the reasons why I want to help restore the southern hard clam population here. The northern hard clam, the reason that they do the aquaculture of the northern hard clams is because they basically just have a longer shelf life than the southern hard clams, so they're safer for humans to consume. But the southern hard clam, since it is a native here, it also grows faster. It's ideal for restoration and ecosystem management. So this image here is actually what an aquaculture farm for hard clams looks like. It's basically a bunch of PVC poles sticking out of the water, and on the bottom is attached bags full of hard clams. So if you see something like this when you're out in the water, that's probably what you're looking at. We do have one in Pine Island Sound that you might have seen before. When scientists looked at the genetics of the hard clams at several sites along the coast of Florida, they actually found that the southern hard clam species were becoming more rare due to hybridization. And this is an issue because the hybrids can actually be slightly more susceptible to diseases. So in order to understand the potential that hard clam restoration would have to improve the water quality and restore about these valuable ecosystem services in Pine Island Sound, we will about 12,000 clams in an area. That's about 13 clams per meter squared. And the reason we want to put so many clams in such a small area is because clams are broadcast spawners. A broadcast spawner basically means that they release their gametes into the water column. So this really increases their chance of fertilization if they're really densely populated in an area. That way their sperm and egg can meet in the water column to produce more offspring. Once we plant the clams out in this area, we're going to monitor their survival, reproduction, and also see if they have any effect on water quality, which would be great if they did, for 1 year. So this is the area where we're going to be planting it around Sanibel. This red zone here is prohibited for shellfishing, and the green areas are conditionally approved depending on water quality problems. They close it down after heavy rains or if there's red tide for neurotoxic shellfish poisoning. We're going to be planting it in the prohibited area, so hopefully that prevents any poachers from taking it, and also we're going to hopefully put them in an area that people can't really access them. So we're going to monitor several aspects of the hard clams to see how they respond to being planted in Pine Island Sound. And we'll first of all be monitoring the survival and growth. We'll be going back regularly to check that the clams are surviving or see what their survival rate is. We'll also be taking measurements of their growth. Hopefully, we do expect them to grow where we're planting them because they will have plenty of food. And we'll also be collecting several of the clams during each trip to study their reproductive cycle and determine their condition index. The condition index is kind of a value that tells you how healthy or how much meat is basically inside of the shell compared to the volume of the shell. And it changes seasonally and with their reproductive status. We'll also be determining be determining the sex of the clams because we want to know the sex ratio. So clams are actually sequential hermaphrodites. So that means that they start off as males and they switch to females when they get larger. So we're starting out with younger clams, so we expect the population to be mostly male at the beginning, and then we'll expect to see more females as time goes on and hopefully see like a 50-50 sex ratio once the population gets established. So that can be a really interesting way to monitor them and see what happens. The gonad ripeness is basically these pictures up here that you see. This is from inside the clam. You open up the gonads and you can see if this picture has nothing in it and this one's full. So this one's gonna get ready to spawn soon. And you can basically tell, you can't be out there every day to see if they're spawning. But if you open them up one day and they're full mostly, and then the next day they're empty, that means that they've had a spawning event. And that means that they're having success out there. Another thing we can look at is for the male to female ratio is, this is how you can tell if they're a male. You take a swab of this, put it under a microscope, and this is the sperm, these little tiny rods, and then the females have these big obvious eggs. So that's another thing we're going to be doing, and just seeing how much sperm and eggs there are. This will help us understand the gonadal cycle of hard clam populations in this region, which is variable across different regions. Sometimes they spawn twice a year, other times they may only spawn once a year, but we don't know that for this region specifically. We'll also be monitoring water quality, and I'll get more into each of these a little bit, but we are going to look at the phytoplankton community structure using our Flowcam, which is a great sort of new or novel tool that we're still learning about in the lab. It's kind of powerful, but we haven't really been able to like capture how powerful it can be. And I think that this project is really going to help us with that. And we're also just going to be taking regular water quality monitoring, things like DO, chlorophyll A, color dissolved organic matter, CDOM, pH, salinity, and temperature. All of these variables are very important for clam survival. And DO is dissolved oxygen. And we'll also be measuring different nutrients to see what effect the hard clams have on the nutrients in the water column where we plant them. So this slide is showing the habitat suitability model that my coworker Mark Thompson just did. So, and it's specifically for hard clams. And he basically took all of these different factors here and made different maps to see if the hard clams could live there. And then he layered them all on top of each other and was able to basically just show using colors where it's good for clams to live and where clams can't live. So green areas are great areas for clams. These red areas are bad for clams to live in. So there's a lot of different factors. Salinity is a big factor for clams. I would say anything between 20 and 35 is a good range for them, but once you start getting into these lower salinity areas, that can become lethal for them. So we don't want anything in this area near the mouth of the Caloosahatchee River. They also really like to live in seagrass. They prefer seagrass habitat. But the one issue that I have with putting clams directly in seagrass when we're doing research is I don't want to disturb seagrass beds. And it also would be more difficult to get a permit to put clams in seagrass beds because we're going to be going out there stomping around. And, as, a conservation foundation, we don't want to be messing up seagrass. We love seagrass at SCCF. So I'm going to be putting them more in an area that has less seagrass. And I think an interesting future research for this would be to see if the seagrass and the hard clams have a mutually beneficial relationship. So it would be interesting to see if seagrass starts to grow in where we've planted hard clams. So not only would it be better for water quality, but hopefully it could help improve seagrass ecosystems. Flow rate's also another important factor for hard clams. You want the flow rate to be fast enough that they're getting access to food. Their food is in the water column, and then they suck up the phytoplankton through something called a siphon. And if the water flow is too slow, that means they're not getting enough food. But if the water flow is too fast, then it's going too fast over their siphons and they can't take it up. The substrate that they prefer is sandy mud or shelly sand to muddy sand. Once you get too muddy, the mud starts to become anoxic and they can't live without oxygen. They prefer areas that are 10 meters of depth or shallower. And for us, we kind of want it to be a little bit deeper. That way people don't accidentally find the site and harvest our clams. And also dissolved oxygen and temperature are really important factors as well. So he took all of these and layered them on top of each other. And now we have a pretty good idea of where a good place to put clams is for restoration, or even looking for clams to see if they're out there. The great thing about clams is they're way more robust to the effects of red tide than scallops are. So hard clams can live for over 20 years if they aren't harvested. And even if they have a bad year with red tide and it affects their reproduction, they'll still be alive for the next year and be able to reproduce again if there is no red tide event again. So that's one reason why I think hard clams are going to be great restoration tools, because they're pretty hardy animals. So we have already gone out and surveyed a couple sites. We have a restoration site and a control site. We just want to have a control site where we don't put any clams, so we can see if the effect that we see in the restoration site is due to just clams or if it's due to anything else. Like if we see a lot of seagrass growing in both sites, then we might think, oh, like maybe it's just a great year for seagrass to go. But if it only is growing in the restoration site, then we can say, oh, the clams are having an effect and they're helping the seagrass grow here, or they're improving the water quality, or there's fewer harmful algae in this site than the other site. So the restoration site was kind of a muddy sand area. It had a lot of tube worms in it, and it had some seagrass. So this graph here is showing the seagrass. It had about 50 shoots per meter squared. The control site had a little bit more. And at both sites didn't really have any predators because, well, I didn't find any clams at either of the sites. So there were, of course, no predators. There was one whelk at each site. And there was macroalgae at both the sites too. They were about 2 meters deep. And these are showing the nutrient samples that we took of the sites. They're pretty low and they're about the same at each site. So I think these are going to be really good sites to compare to each other, and they are adjacent to each other. So anything that affects one site should affect the other as far as water quality is concerned. The Flowcam and phytoplankton. The Flowcam is basically a microscope attached to a camera, which sends pictures to the computer, and you kind of get results like this. And you put the sample through this this little tube, and this sample through, and every time a tiny particle comes picture. It's an amazing tool. This is a shot of what you can get. As you can see, some of these things are almost impossible to identify as phytoplankton, like to the species level especially. But one powerful thing you can do for this project specifically is count the particles per milliliter. So in my restoration site, there started out like 6,300 particles per milliliter. And we would expect that with the hard clams present, we would see a reduction in the number of particles per milliliter. And that would mean that they're eating the plankton and they're allowing more light to penetrate the water for seagrass. That's my hypothesis. This is some pictures from a project that we had an intern do last year. She was able to take a lot of pictures from samples all along the Caloosahatchee Estuary, Pine Island Sound, and the Gulf of Mexico. And we sampled all those sites several days throughout the year. And she grouped them. She wasn't able to identify them all down to the species, but she grouped them by like common features, like these are dots. She said these are dots, these are squares. This is a navicula. You can ID a lot of them. We also have like centric diatoms, pennates, bead chain diatoms, filamentous cyanobacteria, ciliates. Cattosaurus is a really common one that we see, and skeletonema. She found a bunch of different things, and then she made this really amazing, she interpreted this very well. I'll walk through this for you. Basically what you have is all the sites starting up the Caloosahatchee River all the way to the Gulf of Mexico. And these are all the IDs. So she grouped them like tubes with spots, cylindrotheca, serratium, and all those colors match up to these bars. So you can see how the ecosystem, like this is low salinity and this is high salinity, that affects the type of phytoplankton you see. So this is what I plan on doing. I plan on doing something similar to this for phytoplankton community structure with the samples that were taken from the sites before, during, and at the end of the restoration period. So the next steps, and everything's actually going to be happening pretty soon. We need to obtain our special activities license from the FWC. This is taking a very long time. I submitted it in February, but I emailed her last week and she said she's gonna get started on it now, and we should be receiving this. You need a license to plant clams out in the estuary. We should be receiving it in the next couple of weeks. And our clams are actually growing at the aquaculture site in Pine Island Sound. So they should be ready, and they should be fully grown, which will help protect them from predation next month. And we'll just be picking them up from the farmer, Carter Davis. And we're actually working with Sarasota Baywatch. They donated the 12,000 clams that we need to us, which is a really amazing collaboration that I'm excited to have with them. So I want to say thank you to everyone, the Marine Lab, of course, and to Sarasota Baywatch and my other collaborators. And of course, thank you to the Sanibel-Captiva Shell Club for hosting me today and for helping fund this project.
Well, we have one person who just wants to confirm, and I think this is Mindy. She thinks she has, but are these also called Southern Quahogs?
Yes.
Okay.
Yeah, that's what they call them in New England.
Okay, great. I was curious if they spawn all at the same time, or do they have a cycle that they go through like some other species do?
So the way that the hard clams spawn is basically dependent on temperature. And once the males release their spawn, I think it's around 23 degrees Celsius, which is pretty warm, the females notice and then they release their gametes.
And Joe would like to know if the Lake Okeechobee discharges affect salinity or any other aspect.
Absolutely. So remember the graph that I was showing you with the, let's see if I can get back to there, this. So this is the salinity minimum that they can't endure. And basically, if you have too much releases, that salinity minimum is going to stretch out into the estuary, which is something that would be really bad for hard clams planted in this area. And especially if the duration was longer than, you might start to see a drop. So we are going to continue to fight to have them not do those regulatory releases from Lake Okeechobee and just maintain a healthy salinity for our estuaries.
Your video cut out for just a second there. What was the period of time that they could survive a higher salinity?
3 to 6 days, probably.
Okay.
You'd start to see some effects.
Okay. Another person wanted to know if they're how they live in those buckets.
The ones on the last slide.
Yes, I think as you were about to put them in.
Yeah, so they can close up their shells really well and survive for a long time out of the water. They're actually a lot more sensitive to temperature when they're in these buckets like this. So Sarasota Baywatch, they actually have them trucked in these like big Budweiser vehicles that are refrigerated. So that they were actually like Budweiser donated those trucks to them, which was really funny and cool that they were able to get that. So they can survive pretty long time without being in the water.
Great.
But they have to be cold.
Have to be cool. Okay.
Yeah.
Like your beer.
Yeah, exactly.
And exactly what is the process with the 12,000 shells in terms of how do you just drop them over, or what do you do?
Yep. So we're going to put them all in our the Norma Campbell, our research vessel, and buckets just like these. This is a picture from their project. And we're going to just pick them up from where they're being farmed in Pine Island Sound and drop them off at our site, which isn't that far away, and basically just dump them overboard. And then we might have a diver go down and make sure there's no big piles and spread them out nice and evenly.
Okay, great.